Hi, my name is Jose Flores, and this is my video presentation for my case connection questions for medical biochemistry. All right, so to get started, we're talking about the case connection for pyruvate kinase and its regulation and roles in cancer metabolism, and a case study involving insulin and a pediatric patient. Our discussion will cover the normal regulation of pyruvate kinase, its altered function in cancer cells, and what insulin measurements can reveal about a patient's metabolic condition. Let's begin with the regulation of pyruvate kinase under its normal conditions. So right here, I've provided the chain that shows glycolysis, and we're going to talk about the ending of it for a second. In the end, we know that one glucose molecule produces two pyruvate molecules along with a small amount of ATP or NADH, so energy. This process does not need oxygen and is the first step in getting energy from glucose, whether oxygen is present or not. Prior to that, the first case shows that glucose is prepared by adding some phosphate groups to make it split a little bit easier. Then the glucose is split into 3 carbon atoms, or 3 carbon molecules. These smaller molecules go through a few more changes that release energy, and some of this energy is captured as ATP, which the cell uses for various activities, and some is stored in molecules called NADH. In the next slide, we see that pyruvate kinase is a pivotal enzyme in glycolysis, the metabolic pathway that converts glucose into pyruvate, producing ATP in the process. The enzyme is crucial for energy production in cells, and in mammals, there are 4 isoenzymes of pyruvate kinase that are present, and they all are in different specific areas and have different needs of different tissues. So we're going to talk about type L, which is found in the liver and kidneys, and it plays a role in regulating blood glucose level by adjusting glycolysis and gluconeogenesis based on hormonal signals. Type R, the second one, is expressed in erythrocytes, so red blood cells, and it supports anaerobic glycolysis and is essential for lack of mitochondria cells, or essential for cells with lack of mitochondria. Type M1 is the third one and is present in muscle and brain tissues. It supports high energy demands during intense activity. Number 4 is type M2, found in self-renewing cells like stem cells and various cancer cells. It has a unique regulatory role crucial for cell proliferation. Pyruvate kinase is regulated through key mechanisms, and one of them would be the activation of allosteric regulation. This is when the fructose 1,6-biphosphate, a glycolytic intermediate, binds to pyruvate kinase, enhancing its activity. This ensures that the enzyme operates effectively and efficiently when glycolysis is active. The inhibited state with ATP acts as an allosteric inhibitor and signaling that the cell has sufficient energy and glycolysis should slow down and conserve resources. Covalent modification is another key mechanism, and it's in the liver. Pyruvate kinase type L is phosphorylated by protein kinase A, or pKa, in response to glucagon during fasting. Phosphorylation decreases this activity, thereby reducing glycolysis and promoting gluconeogenesis to maintain blood glucose levels. Isoenzyme-specific regulations, like type M2 in cancer cells, is regulated differently, and it often exists in less active form, which allows for the accumulation of glycolytic intermediates that can be diverted into anabolic pathways necessary for rapid cell growth. I will continue to talk more about type M2 and its regulations. Inputted right here, we could see a normal cell and a cancer cell with pyruvate kinase type M2 and the differences between these two cells. When pyruvate kinase type M2 is notably different in cancer cells compared to its counterparts, it is often found in low activity states, which is counterintuitive for glycolytic tissues that require high enzyme activity to drive glycolysis. The rationale for this is the Warburg effect. The Warburg effect is when cancer cells exhibit increased glycolysis, even if it is in the presence of oxygen. Pyruvate kinase M2's low activity supports this by allowing glycolytic intermediates, as said prior, to accumulate and be diverted into biosynthetic pathways, crucial for rapid cell proliferation, once again stated before. Metabolic adaptation is another rationale. The shift in metabolism supports the production of nucleotides, amino acids, and lipids necessary for the formation of new cells. In the next case connection, we're going to talk about a case involving a 5-year-old boy with persistent thirst, frequent urination, and drowsiness. His insulin levels were measured at the baseline and 1 hour after a glucose loading test. And these are the results. We could see that for the serum insulin levels in milliunits, the base bloodline sample with the patient is at about 0.8 with a control of 6 plus or minus 2. 1 hour after the glucose load shows that the patient has the millionit of 5 insulin level with a control of 40 plus or minus 11. These measurements provide insight into the boy's insulin production and response to the glucose intake. For the introduction of the case study, we can see that in the insulin levels, the patient's baseline insulin level is significantly lower than the control, which indicates a deficiency in insulin production. After the glucose intake, the patient's insulin response is much lower than the control, suggesting impaired insulin secretion or beta cell function. These findings are consistent with type 1 diabetes, characterized by autoimmune destruction of insulin-producing beta cells in the pancreas. Predicted fasting blood glucose levels show that the low insulin levels and impaired response. Given the low insulin levels and impaired response, the patient's fasting blood glucose levels are likely elevated, consistent with hypoglycemia seen in diabetes type 1. I've provided a graph to show the x-axis in time, baseline and 1 hour after glucose load, and the y-axis being the serum insulin levels in milliunits. And the two lines indicate the patient, which shows a low baseline level with a modest increase after glucose intake, and the control, which displays a higher baseline level with a significant increase post-glucose load. Once we analyze the insulin levels, we can see that in type 1 diabetes, the following regulatory enzymes are in particular interest. We're going to talk about hexokinase, and the function of hexokinase is that it catalyzes the first step of glycolysis, converting glucose to glucose 6-phosphate. In response to high blood glucose levels, hexokinase activity would increase to facilitate glucose uptake into cells. The second one would be glycogen phosphorylase, which is involved in glycogen breakdown to release glucose into the bloodstream. Elevated glycogen phosphorylase activity would help to manage high blood glucose levels by increasing the amount of glucose released from glycogen stores. These enzymes help in managing glucose metabolism under conditions of impaired insulin signaling by promoting glucose uptake and release. Once we understand the regulation of pyruvate kinase and its role in cancer metabolism, it highlights the complexity of metabolic pathways. For the case 1, in cancer, the unique regulation of pyruvate kinase M2 not only supports rapid cell growth, but offers potential therapeutic targets. Targeting pyruvate kinase M2 or its regulatory pathways could provide new strategies for cancer treatment. For the second case connection of the patient, in type 1 diabetes, we get a better understanding of insulin dynamics and a role of key enzymes like hexokinase and glycogen phosphorylase, and can aid in developing more effective treatments and management strategies for this type of diabetes. In conclusion, pyruvate kinase regulation involves complex mechanisms, including allosteric regulation, covalent modification, and isoform-specific adaptations. In cancer, pyruvate kinase type M2 supports the Warburg effect and redirecting glycolytic intermediates into biosynthetic pathways. And the case study of the pediatric patient underscores the importance of understanding insulin dynamics and regulation in managing type 1 diabetes. Here are some references. The first case connection was very baseline and crossed paths with a lot of molecular biology and biochemistry. So I have inputted Albert's B, molecular biology of the cell, and biochemistry from Berg. And for the second case connection, we're going to look at the American Diabetes Association and the diagnosis and classification of diabetes type 1, and diabetes cared, both of which show examples of insulin levels and their changes when it comes to inhibitors. Thank you.